My name is Boris Rudolphs. I am an undergraduate in biochemistry at Fresno State, and I'm here to talk to you today about fast computational design of riboswitch complexes using domain matching strategies. So I didn't do too much of a background slide or anything like that, but a short discussion about Eterna switches. So Eterna has come quite far. We've done computational design of in-state puzzles and sequences, the arithmetic puzzles, OpenTB, and OpenCRISPR and others puzzles as well. And this is just a random puzzle that had two solvers that I'd solved, I don't know, a couple of years ago that was incredibly difficult. And it's just like, wow, look how far I've come. So I asked some players to give me their switch design strategies because I was going to include a couple of quotes in this presentation. In this case, I didn't include their names because I forgot to ask for permission on that, but they essentially describe the way that they solve switch puzzles, especially in the labs, is they just try to match sequences to usually the MS2 hairpin or possibly a signal sequence. And as an example, sometimes I paste a second MS2 around until it snaps together with the first one. Again, complementary sequence to the MS2 hairpin, just coming together. And then the second quote usually starts with identifying shared or complementary sequences. Yeah, perfect. And yeah, so that's pretty much what this strategy that I developed for originally XOR puzzles was. But first we'll go with the signal structure relationship. And for any RNA sequence that acts as a signal within a nucleic acid, there are two properties. And the first is that the sequence associated with that signal will adopt some stable conformation. As an example, the MS2 hairpin protein complex or an oligo-bound nucleic acid dimer. And the second property, to turn off that signal, some modification will need to be made to that stable conformation, such as a chemical modification or a catalyzed cleavage, such as a ribozyme or a protein enzyme, and then possibly just additional structural changes due to thermodynamics and changes to the equilibria. And so domain matching secondary structure design, DMSSD, it takes the secondary structure of switches and it reduces it to essentially a string of characters. In this instance, I have an FMM riboswitch, just two secondary structures that is reduced to a series of functional domains. First being the N1 domain, the FMN1, FMN2 domain, which is represented by these sequences and their adjacent helices. A shared FMN2, MS2-1 domain here and here. The MS2-1 domain, the MS2-2 domain, an FMM1 prime domain, which I believe is this. And then you have this N3 sequence, which is essentially just unpaired bases. And then you have FMN1 double prime, where this sequence here is complementary to this sequence here in this region. And then you have MS2 2 prime. So here, this is going to be complementary to, I believe, somewhere around here. And then you have the N4 domain, which is complementary to the N1 domain. And then you can also include the base pairing between these individual domains as well, if you want to further compact it. And so we originally used DMSSD for the XOR puzzles, and the XOR puzzles were arithmetic puzzles that had the MS2 hairpin as a signal, where in order for the signal to occur, you had to fold one, the hairpin must fold when one or of either two oligonucleotide inputs bind to a single sensor RNA. And it also has to occur when both or neither oligonucleotide are present, the MS2 hairpin must not form. And so apparently this was quite difficult. From what I understand, the DOS lab spent hours working on a solution, and I believe Nando managed to get a solution like 6 hours later or something like that. And as it turns out, there's actually a solution, an analytical solution to this problem, where your first oligonucleotide binds to this O domain here. Your second oligonucleotide binds to this X domain here. you have this N1 domain, which is complementary to both the MS2 protein hairpin domain and as well as this N3M domain here. And then you have this X domain, which is complementary to the second oligonucleotide, is also complementary to this N3X domain. And likewise, there's an N3O domain complementary to the O domain here. And as it turns out that when the first oligonucleotide is bound, your MS2 hairpin will form because the N3X domain binds here, preventing the N3M domain, which should bind to this M domain, from occurring. Likewise, when just the second oligonucleotide is bound, that releases the N3X domain, and the N3M domain binds to N1. And then when both of them are bound, the M domain is going to bind with the N3M domain. And when neither of them are bound, the the N1 domain should bind to the M domain. So you get this analytical solution that is completely input independent, as it turns out. And we essentially decided to automate making solutions for it. And the way that went about is that Nando wrote the code for this, and if it solved it, he kept the solution. If it didn't solve it, he just started it over. There was no optimization of sequences at all. And so as you can see from the round 98 results, average scores for the DMSSD algorithm were quite high. And in fact, the highest score was from this algorithm. And a lot of player sequences were modifications of sequences that this algorithm made. And likewise, in round 102, same continuation of the logic gate labs, we had the XOR puzzles again. And players, they took the DMSSD sequences from the previous round and they optimized it to reach almost perfect scores. And additional solutions were submitted from the DMSSD algorithm, and those scored almost a little bit lower than 90 or so. Nando also coded something for the XOR logic gate puzzles, which just took the similar idea of using domain matching strategies to create solutions. And it didn't do that well. I think we ended up determining later that his code did something that wasn't similar to what the strategy entails. So it kind of tells you a little bit about how effective that strategy was for the XOR puzzles, but maybe not so much for other sequences or other puzzles. But let's continue on. And here's this beautiful graph showing full change with respect to the location of each of these individual domains. And here we have the MS2 domain for a number of the high scoring sequences, an oligo binding site for both the first and the second. And as you can see, a lot of these sequences are very similar. They have very similar domain locations, very similar probably sequences to the extent that the MS2 hairpin and these binding sites for the oligonucleotides are fairly well conserved. And from there, we decided to check in silico these XOR solutions. And with the experimental evidence being a success, it was repeated with another motif to act as a signal and the combination of 6 different microRNA input molecules. And we used the kink turn, which is an internal loop, as the signal. And we ended up using the first 6 or all 6 of the oligonucleotides from the TB puzzles as the inputs. And as you can see here, as an example, A1B128, that was the number of iterations that was required to reach a solution. So 28 seconds or so after starting the algorithm, it came up with an analytical solution. And then it was a success for every single input, essentially showing that we had created an input-independent XOR solution. And from there, we decided to try and apply this to OpenTB. And I identified 3 strategies for doing this. You may recognize this first one here. It's actually WH Bob. It's the same exact base pairing strategy, right? Where C1 and C2 are essentially connected to R, and you have these complementary domains that bind to your reporter oligonucleotides sequence in the presence of C1 and C2. But when A and B are present, it shouldn't bind. And these are actually the original screenshots I took of this to give to Nando to say, hey, let's turn this one into a strategy. And we decided that this one, there are a couple of issues with it. For example, the fact that A and B have to bind to this, essentially an open hairpin, suggested that it may not actually act as a switch. It could just bind to it, and we wouldn't know if it moved in one direction or not to a certain extent. This second strategy is actually quite complicated. Your A and B oligonucleotides bind here, dissociating this helix, releasing the CCR complementary sequence to bind to R sequence here, leaving these two sequences to bind up with possibly C1 or C2. And it required 8 functional domains, which in order to bind the oligonucleotides, you need about 10 or so minimum. So you start to run into a size limit as a result. And it actually turns out that we went with another complicated one as a result of all that, because this one just apparently seemed better than the rest. So Nando implemented strategy 3 within DMSSD. I submitted a number of solutions from strategy number 1, and I believe I tried a couple from strategy number 2. And as you can see, there was a continued success for DMSSD in the OpenTB results. I would like to note that these are a graph of the Eterna score with respect to the DMSSD and the player solutions. And it showed higher average and median scores compared to players. And we have seen that there are a couple of players who have fantastic designs, such as Andrew and GL2007. And it's quite amazing, like what those individual players have done. But this strategy here, it being input independent, could possibly just allow you to, if you had a similar system, to use those oligonucleotides to design that. And yeah, so future and conclusions of DMSSD, successful strategy for identifying riboswitch architecture for design input, to design input independent RNA sensors. Future developments include automating this process. All the strategies, I had to essentially sit down with a piece of paper and start connecting domains in order to see if, how does this base pair, this helix here, does it break? Does it retain its structure when oligo A or oligo B binds? And it should be interesting to automate this process, because we could use this for essentially any RNA sensor. And expanding this method to include the structures of competitive binding solutions, similar to Andrew's. One issue is that when you're using DMSSD, all your inputs are independent, so they don't competitively bind to the same exact sites, correct? And because that occurs, to a certain extent, I do believe that for some of the more complex problems, like let's say 6 inputs, you may not be able to get an analytical solution with a length of 80 nucleotides. It may actually be impossible, at least for input independence. And I also want to quantify the Eterna data set and take that information to optimize solutions designed by this strategy. None of these designs were optimized. It was just Nando ran it. If it worked, it worked. If it didn't work, I can't remember how long it takes. It took a couple of minutes. Then it would just go on and continue its iterations until it found a solution that worked. And I think that's also another important point. Let's go back to the XOR. This takes a couple of seconds to generate a solution. For players, on average, it took me around 15 minutes or so to design a solution using this same exact strategy. With automation comes certain degree of efficiency and a certain degree of speed that is really important for this kind of application. And so moving towards automated design algorithm that identifies the inputs and signals, identifies any potential architecture that could solve this puzzle, and designs a sequence that fills the secondary structure architecture and refines solutions that fulfill these requirements. I would like to acknowledge Fernando. He's amazing. Without him, none of this would have probably gone anywhere. I'd also like to thank Dr. Riju Das, the DOS Lab, Eterna players, faculty from Fresno State, yeah, all the people who've helped me over the years. And yeah, any questions?